Hello, and welcome back to Mammalian Anatomy at Keuka College. In this video, we're going to be continuing our series on chapters 19 and 20, the heart and blood vessels, where we'll focus on the anatomy of the heart and blood vessels. And in this video, we're looking at the conduction system. We're not going to get into the nitty-gritty details of ion channels and things like that, so we're just going to introduce it. But I have to say, this is one of my favorite parts of the heart. It is so cool. Let's get into it. So, did you know that the heart can actually generate its own heartbeat and actually control its own heart rhythm? No nerves, no hormones, nothing, just all by itself. And that is super, super sweet. So you could actually take the heart out of the body and it'll continue to beat. So cool. So we call this ability to generate its own heartbeat, or more specifically, to depolarize and create its own action potential, automaticity. So to be able to generate your own action potential and depolarize yourself or the heart itself is automaticity. Autorhythmicity, on the other hand, means to generate its own heart rate or heartbeat. It controls its own rhythm. So we have two really related words, but the heart really can generate its own action potential and control its own heart rate. So that is really awesome. So this whole entire process is going to be accomplished by the conduction system. So what is the conduction system? And let's talk about it together. So the conduction system is composed of specialized cells called conduction cells, or I may also call them pacemaker cells. And these cells are really important because they are able to generate their own action potential. So because of the way their ion channels are set up, it can actually stimulate itself to threshold and cause the cell to depolarize, just like a nerve can depolarize and have an action potential. These cells can also generate an action potential and have an electrical impulse go through it. It can do, and these cells can just generate their own and make their own action potential. So they don't need to be stimulated by a different stimulus or by a nerve or hormone. They can just generate themselves. So, it is really, really cool. I'm trying not to go deep into the physiology because I could talk about it for hours, but we'll get into the physiology of it in vertebrate phys next year. So all the channels, all the ion channels, get hyped for the ion channels. Okay, but let's talk about the conduction system in general. What are its functions? The function of the conduction system is 1, to generate an action potential, 2, to propagate or move that action potential through the heart, 3, stimulate the cardiac muscle cells to contract, and 4, coordinate the contractions of the atria and ventricles. And so it's really, really cool. And this is where that action potential is going to propagate through those gap junctions, those electrical synapses. And because each of the cells are connected using an electrical synapse or using those gap junctions, the action potential will travel through and sweep through the heart, and it will stimulate the contractile cells or the muscle cells to contract and coordinate the contraction of the heart. So cool. Sorry, geeking out so much. All right. So first and foremost, let's go into kind of the components and label the different parts of the conduction system. And then what we'll do is we'll look at how the electrical impulse will actually travel through the conduction system. The first part of the conduction system is the sinoatrial node over here. And the sinoatrial node is important because it is the primary pacemaker cells, or the primary conduction cells. This area is going to generate the action potential that is going to travel through the rest of the conduction system. Do note, other regions such as the AV node and the Purkinje fibers can also generate an action potential, but the sinoatrial node generates the action potential at the fastest rate. So it's going to override the action potential made by the AV node and the Purkinje fibers. So for our purposes, the SA node will generate the action potential. That SA node is going to go through specialized areas called the internodal pathway. So it's going to travel from gap junction to gap junction, from pacemaker cell to pacemaker cell, and it's going to travel through the atria, so both sides of the atrium, right and left atrium. And as it does this, it's going to stimulate the contractile cells, or the muscle cells of the atria, and stimulate them to contract. Over here we have the AV node. The AV node is where all of those signals are going to converge. So it's going to, the action potential is going to start at the SA node, sweep over the atria, and then converge at the AV node. So start at the SA node, sweep over the atria, converge at the AV node. This AV node is really important because the action potential travels very slowly through the AV node, or the atrioventricular node. And this slow motion is essential because it allows the atria to fully contract before the ventricles begin to contract. If it didn't travel slowly, the atria and the ventricles would contract at the same time, and the heart wouldn't be able to fill and pump blood effectively. Because of this pause, the atria can fully contract, filling the ventricles up, and then the ventricles will contract, pumping as much blood out as it can for that heartbeat. So remember, the AV node creates a pause. The electrical signal then will pass through the AV bundle, or I may also call it the bundle of His. I learned a lot of the older names, so sometimes what I'll have is the older names in gray. So if I use AV bundle or bundle of His, know that I'm referring to the same structure. Over here, what we're going to have is from the AV bundle, the electrical impulse will travel through the bundle branches, and this is going through the septum of the heart. It's going to go between the ventricles, and it will loop around to the Purkinje fibers over here. There's a new name for the Purkinje fibers, but it is longer than it has to be, so I'll just stick with Purkinje fibers for now. So the action potential will go up here, and what will happen is as it's looping around, the Purkinje fibers are going to stimulate the walls of the ventricle to contract. So we have the SA node going through the internodal pathway, taking a pause in the AV node, coming down the AV bundle, going through the AV bundle branches, stimulating the septum of the ventricles, and coming up and looping around the Purkinje fibers, and this is going to stimulate the wall of the ventricles. So let's look at kind of the steps. So what will happen at time 0 is the SA node will generate the action potential, and that and that action potential will go through the internodal pathways, spread through the atria, stimulating the atria to contract. So over here, you can see the SA node starts here at time 0, and like at time 15 milliseconds, we can see that the electrical impulse is spreading across the atria, and it'll eventually meet up with the AV node. At time 150 milliseconds, what will happen is that there's going to be a bit of a delay between the stimulation of the atria and then the contraction of the atria. Electrical impulses always come first, then mechanical action. So you got to stimulate the muscles to contract before they contract. So here we can see that they're contracting. And over here, there's going to be the AV node. The AV node is going to create a little bit of a pause, allowing for the atria to fully contract before it's going to allow for the stimulation and contraction of the ventricles. Here we're going to see that the electrical impulse is going through the AV bundle and AV bundle branches. When it goes through the AV bundle branches, the electrical signal is actually stimulating the septum of the ventricles to contract. And then what will happen is the electrical impulse will come up the Purkinje fiber and stimulate the ventricles, or the muscles of the walls of the ventricles. And this will help stimulate the ventricles to contract. So SA node, atria through the internodal pathway, AV node, AV bundle branches, Purkinje fibers, and the ventricles. And remember, there's always going to be stimulation of the cardiac muscle tissue, then contraction. They're separated from each other. That way, there's going to be a bit of a delay. So those are the steps of the conduction system. What I'd like you to be able to do is be able to describe in an essay format the steps involved in the conduction system. An electrocardiogram has a capability of graphing the electrical impulses that go through the heart, and we may call it an EKG or an ECG. You may see it as EKG because electrocardiogram actually came from Dutch, and it's spelled with a K instead of a C. So essentially, what is an electrocardiogram? Let's talk about it. So when the heart is actually having these electrical impulses will actually loop around and come to the surface of the skin. And we can actually place electrodes onto the surface of the skin to be able to detect these electrical currents. And so here we're looking at a 12-point EKG that is put on the patient's chest. That is going to be much more specific. And what will happen is we'll be able to see the different phases of the electrical impulse, and we'll get a graph that kind of looks like this. We'll have the P wave, the QRS, and the T wave. I'm not going to make us go into every little detail or understand the specific physics behind the electrocardiogram. Mainly what I want us to learn is, what does the P, QRS, and T wave represent? So the P wave represents the atrial depolarization. It essentially represents when the electrical impulse is going through the atria and making the atria positively charged. So this is when the electrical impulse is going through the atria. The QRS is representing when the electrical impulse is traveling through the ventricles and making the ventricles positively charged. Also, during this time, the atria are going back to normal. They're going to repolarize and go back to being negatively charged. So we have the P wave and the QRS wave. This is going to be depolarization of the atria and depolarization of the ventricles. And then over here, we have the T wave. The T wave is representing the ventricles going back to normal, going back to being negative. So P wave represents ventricles, and P wave represents the atria becoming positive. QRS represents the ventricles becoming positive and the atria becoming negative, going back to normal. And here we have the T wave, which represents the ventricles going back to normal, going back to negative. We'll get into electrocardiograms in much more detail next year when we're in vertebrate physiology. We'll look at how the electrocardiogram can actually pick up these signals and understand a little bit more detail what I mean by positive and negative. So here, just know in a general sense. And we'll also be able to do our own EKGs. We're going to do a 3-point EKG rather than a 12-point. So the the EKGs will only be on our wrists and our ankles rather than across our chest. One caveat, even though the heart is an amazing organ and it can produce its own heartbeat and it can set its own rhythm, the heart needs to be modulated by the nervous system and hormones, specifically to regulate blood pressure in the body. And so the heart is going to be innervated by the sympathetic nervous system, that fight or flight nervous system when we get nervous or when we're in a dangerous situation and our heart is beating faster. And it's also going to be stimulated by the parasympathetic nervous system, that rest and digest, relax and slow down kind of system. So the sympathetic nervous system is going to be our fight or flight, and this is going to help us deal with dangerous situations, nervous situations, or even if we're just working out. And what will happen is this will actually innervate the conduction system, so like the SA node and AV node, and it will also innervate the atria and ventricular myocardium, the muscles of the heart as well. Because it innervates the conduction system, it's going to speed up the heart rate, so it's going to make our heart beat faster, help increase our blood pressure and stuff like that. And also because it innervates the myocardium, the muscle, it's going to increase contractility or the strength of contraction. So not only is our heart going to beat faster, it's going to be harder and squeeze harder as a result. On the flip side, we're going to have the parasympathetic nervous system. The parasympathetic nervous system is going to be kind of the rest, digest, relax, slow down kind of nervous system. And this is going to innervate the SA node and the AV node. So it's going to slow down the heart rate, and it's also going to innervate the atrial myocardium. Not so much the ventricular myocardium, so it's not going to affect contractility as much. So rest and digest. So its major job is to slow down heart rate. So we can kind of see some of that innervation points here. Before we go on, I wanted just to tell you a cute heart pickup line. I think it's adorable. Usually when I'm teaching about the heart, it falls along Valentine's Day, so I think this is kind of cute one to say. There are different chemicals that can actually speed up and slow down heart rate. They are called chronotropic agents, chrono meaning time, tropic means to generate or to make. And positive chronotropic agents speed up the heart rate, and negative chronotropic agents slow down the heart rate. So the joke is, it's like, hey, are you a positive chronotropic agent? Because my heart is racing. So that's a cute one too. All right, well, just something a little extra and fun for the lecture. I think that's it. So that covers the conduction system. In the next part, we're going to kind of start blood vessels and things like that, I think. Yeah. And I will see you next time. Have a great day, and bye-byes.